All right, let's get started. Welcome to today's Molecular Biosciences seminar, which is co-sponsored by the Physical Sciences Oncology Center. Our speaker today is Martin Hetzer, who's coming to us from the Salk Institute. A little background about Martin. Martin grew up and did his undergraduate and PhD degrees in Austria. There he worked with Rudolf Schweyen and did some very interesting work on self-splicing group 2 introns. During his postdoctoral work, he moved to EMBL, where he worked with Ian Matai, and did some very important work trying to understand how both nuclear envelope and nuclear pore complexes are assembled after mitosis. And then in 2004, he moved as an assistant professor to the Salk Institute, and has since been promoted to full professor. And he has done a lot of really exciting work as an independent investigator, starting with trying to understand the question of how nuclear envelopes are formed and remodeled after mitosis, also how nuclear pore complexes assemble, but also moving into sort of different directions. Recently, trying to understand how aging affects nuclear transport, and also how nuclear pore proteins affect chromatin and transcription. And so he has been the recipient of a number of awards. I won't list them all, but I'll just mention that he was a Pew Scholar. He was awarded the ASCB Early Life Scientist Award, and most recently he was awarded the Royal Society Research Merit Award. And so with that, I'll hand it over to Martin, who's going to tell us about the life cycle of the cell nucleus.
Thank you, Jason, for the introduction, and thank you all for coming, bringing your lunch. I'm hungry. Yeah, can someone throw a chocolate Coke at me? That would be great. No, it's okay. Okay, so as the title of my seminar indicates, or at least that was my plan, that my lab is studying the cell nucleus, and in particular, how the organization of the nucleus changes during development, disease, and aging. And when I started my lab, we were mainly focusing on questions how the nuclear membrane forms around chromosomes as they come out of mitosis. And as Jason pointed out, we also studied how nuclear pores are assembled. Those are the transport channels that mediate nucleocytoplasmic transport. And so over the years, we got more and more interested in the role of the nuclear membrane, which you can see nicely delineated the nuclear interior from the rest of the cell, and its role in organization of the genome and also in transcription. And so I will, before I go into the experimental part of my talk, I want to give you a little bit of a background of the nuclear membrane, most of the things that you already know, but I try to develop some of the ideas that we are trying to get at in the lab. And so you all know when, during evolution, the genome was enclosed by a membrane that was a pivotal moment that allowed eukaryotes to develop multiple and sophisticated levels of gene regulation that ultimately led to the formation of multicellular organisms such as us. And so I think there are several key advantages that eukaryotes can take advantage of having the membrane or the genome separated by a membrane. And the first big advantage is, almost a trivial one, is obviously that the genome functions can operate under sort of the protective shield of the nuclear membrane. And that is important, will become important in the last part of my talk. And I just want to point out that obviously the nuclear membrane separates cytoplasmic components, such as mitochondria, from the genome. And obviously, as you know, mitochondria not only produce ATP, but they also produce free radicals, which are mutagens. So it makes a lot of sense to actually separate those organelles from the nucleus. And I will come back to that very point at the end of my talk. Now, the other aspect of having a nuclear membrane is that it acts as a selective membrane barrier between the two compartments. And that is true not only for organelles, but also at the protein and the molecular level. And nuclear pores are those channels I was talking about already. they mediate all active and signal-dependent transport of molecules across the membrane. So every molecule that has to go to the nucleus is obviously translated in the cytoplasm, but has to be imported, and likewise, every RNP complex, every non-coding RNA has to be exported if it has its function in the cytoplasm. And so that was kind of the classical view of the role of the nuclear membrane for eukaryotic cells for a very long time. But then about 10 years ago, or exactly in 1999, so 11 years ago or 12 years ago, that view changed or was modified by the finding that there are proteins that specifically localize to the nuclear membrane, and that seemed to be very interesting. And the first report was a protein called emerin, which is a transmembrane membrane protein that sits in the intranuclear membrane, and that, when mutated, gives a human disease. And since then, we have, from various proteomic but also genetic studies, we know that there are probably more than 100 different proteins that specifically localize to the nuclear membrane, and specifically to the intranuclear membrane, that when mutated or mislocalized cause a vast majority of human diseases, which range from muscular dystrophies, neurodegenerative disorders, to progeria, which is a premature aging disease. So that sparked a lot of interest in the nuclear membrane, and I want to finish this part by saying I think you will hear much more about nuclear membrane proteins and their role in development and disease in the years to come. So my point being that studying the nuclear membrane is not only an academic exercise, but also has some potential clinical relevance. And I want to show you another example why I think studying nuclear organization and function is actually worthwhile. And my favorite example to illustrate that point is actually a discovery that was made more than 150 years ago by a person called Lionel Beale in the King's College Hospital in London. And he was the first one to realize that cancer cells have often very highly abnormally shaped nuclei. And here's one example of a Hodgkin's lymphoma nucleus, and you can see when you compare the nuclei to normal lymphoid cells, this nucleus is highly abnormal. And there are two striking consequences of those findings, I think. First of all, that clinicians use this kind of information to actually tell you what kind of cancer you have. So it has predictive value. But the other thing is that in basic cell biology, these nuclei are treated as if they were normal. And I think it's time to actually try to study what happens, is the biology of those nuclei, and especially the nuclear membrane, under conditions such as this extreme case of cancer. Is this the same? Can we assume that? And I will come back to that in the second part of my talk. So the basic outline of my seminar is I will talk about nuclear pore proteins and their role in gene regulation, which is in differentiation and gene regulation, which is potentially a transport-independent function of those proteins. And then I'll talk about the loss of nuclear permeable membrane integrity in aging and disease. And so to start the first part, I have to introduce the nuclear pore complex, which is, as I said, this essential transport channel that mediates all nuclear cytoplasmic transport. And you really don't have to memorize all the components. I just want to point out it's one of the largest, if not the largest, transport channel. It's made of 30 different components called nucleoporins, or abbreviated Nups. The number is usually referred to the molecular weight. There are only 3 transmembrane components which seem to anchor the pore in the membrane. I want to point out again the nuclear membrane is a double lipid bilayer, so it has an outer membrane which is continuous with the ER, and an inner nuclear membrane which has all these interesting proteins. And I will start by talking about this transmembrane nucleoporin GP210, or Nup210 as I will call it. And that's an interesting one because it has a very large luminal domain. So most of the protein, more than 90%, actually sit in the perinuclear space. It has a single transmembrane domain, and it's only 50 amino acid that actually extends into the pore. So it provides some interaction with nuclear pores. And the general view of what this nucleoporin does is that it's essential for pore assembly and fusion of outer endonuclear membrane. And I will show you some of the evidence that this doesn't seem to be the case, and show you some data that actually suggests it has a completely different role, which we don't know yet, but I think it will, I will show that it's directly linked not to cell division, but to cell differentiation. And so we were studying the expression of nucleopore components during various differentiation processes. And one of the systems we have established in the cell is a muscle differentiation system based on a C2, C12 cells, where you can differentiate myoblasts, so these are dividing cells, into myotubes by simply depleting the serum. And while most nucleopore components are either continue to be expressed or slightly downregulated, Nup98, as you can see here, was strongly upregulated during differentiation. And here you see the mRNA levels, here you see the protein levels. And it's actually interesting, MyoHC, which is obviously a muscle cell specific marker, comes actually on before the bulk of Nup98. And here's a loading control. So the striking thing is that, obviously as a nucleopore component has been identified, so we wanted to know, well, is it really at the pore? And this is simply to show that it is. So we co-localize with MAP4 and 4, which is a classical nucleopore marker, and we see nice co-localization. Also, if we isolate various cellular components, we always see Nup98 together with other nuclear membrane compartments in the nuclear membrane. So it really is incorporated and targeted to the nuclear pore. And this is very differentiation specific, is shown here. So here we have myoblasts. You see here the DAPI stain. They're not MyoHC yet, obviously. They're undifferentiated state. And if we stain for Nup98, we don't get a signal. So the protein is not there. But then we differentiate them into myotubes, MyoHC positive, and now you see this protein being present. So it's induced during a differentiation process. And other transmembrane components like POM1 and all the other ones we tested are obviously present in both. So that's actually the first differentiation-dependent change in nucleopore composition, right? And that already, the absence of both mRNA and the protein in myoblasts, which are dividing cells, already strongly argues that this protein cannot be essential for pore assembly, as apparently in these cells, nucleopores can assemble without it. Now the question, obviously, is it essential for differentiation? And so we knock down Nup98 by siRNA, and you see we get a very decent knockdown. And the striking finding is that here, while you see in control, scrambled muscle cells get nice differentiation, MyoHC positive, multinucleated nuclei. In the absence of Nup98, we don't form myotubes. So cells can exit the cell cycle. They express MyoHC, so they start the differentiation process, but they cannot fuse. So there's no fusion under this condition, which is a very curious stop in a very specific differentiation effect. Well, if it's specific to Nup98, well, we rescue with a GFP tagged Nup98 GFP construct, and see here again, we look at GFP, MyoHC, and then you see in the knockdown, we completely block it. But if we rescue with a heterologous RNA-sensitive mutant, we strongly recover the ability to differentiate, and that's quantified. And when we measure the fusion index, so measure how efficiently fusion occurs, we actually, in the presence of overexpressed Nup98, we actually see it's more efficient. And I think what I just said, cells leave the cell cycle. Most mutations so far actually have a problem in myoblasts leaving the cell cycle. But in this case, they don't. They leave the cell cycle. They start the differentiation process, but they simply don't fuse. And Nup98 is also required for myotube survival. So if we infect with the virus the formed myotubes, they die within a day. So they undergo apoptosis. So it's also Nup98 that is not only required for the differentiation, but also maintenance of these myotubes. And here's the point that I wanted to make. By overexpressing Nup98 already in myoblasts, before differentiation starts, you get a strong stimulation of cell fusion, what you saw in the graph. But here you see like one of these very large abnormal muscle cell that has hundreds of nuclei. The Nup98 is not only expressed in muscle cells. So here I've shown you now there's the first differentiation-dependent difference in nucleopore composition. And so we ask, well, is Nup98 also expressed in other tissues? And here is from the mouse brain atlas, the Allen brain atlas in situ hybridization in different brain regions, and I think you can nicely see that Nup98 is not expressed in all regions in the brain, but specifically in places like the hippocampus and other areas that actually exhibit adult neurogenesis. So it's very interesting areas. So we ask, well, is Nup98 also critical for other differentiation processes other than muscle cell differentiation? And so we differentiated embryonic stem cells into neuronal progenitor cells and then motor neurons. And here are all the controls. ES cells do have a stem cell marker, but don't express nestin. And tubulin B3 is then a variant of tubulin that is specific for fully differentiated neurons. And when we knock down Nup98, as you can see here, we strongly inhibit also the formation of neurons. So Nup98 is not only required for muscle differentiation, but also for the formation of neurons. So it seems to be a more general differentiation factor, but not a general in a sense. There are, for instance, adipocytes, so fat cells don't need Nup98. So it's not every differentiating cell needs it. Okay, so that obviously comes to the question of what does Nup98 do? And obviously, as a nucleopore component, the first thing we thought, well, maybe it is involved in transport. Although that seems not a very likely possibility to begin with, because I told you it's this very, very short 50 amino acid residue that just allows Nup98 to be anchored to the pore. And so we measured transport efficiency in myoblast myotubes in the presence, absence of Nup98, and we cannot detect any defects. As I said, most of the protein is actually in the lumenal domain of the R. And so we wondered, well, is there anything that we can do to understand its function? And so we still don't know it, but what we found by asking the question, well, in the absence of Nup98, are there specific genes that are no longer expressed, and doing microarray studies, we found a class of 300 proteins roughly that are either specifically up or down regulated in the presence of Nup98. And among those 300 proteins, there's an overwhelming, so if you do a Go analysis, there are a large class, and many of them are not characterized yet, large class of proteins that have been implicated in various differentiation developmental processes. And I just show you one of those, GDF5, which is actually has its name, general differentiation factor 5. When we look at this, it's actually expressed after Nup98 comes on. And then if we delete, or if we knock down GDF5, we also block muscle fusion. So we have identified targets that somehow are regulated by GP210. So it's either that GP210 has a very, very specific role in transport for this large class of genes, or that GP210 has something else. And we're obviously trying to figure out what those functions might be. And we think it's a very upstream event of this differentiation process, because if we overexpress Nup98 already in myoblasts, where it's normally not expressed, GDF5 and many of these other targets are also upregulated. So you get even outside of the context of differentiation, GP210 somehow triggers the expression of those genes. It's very curious, and now we have to fill in the puzzle. So how does GP210 do that? And so to complete that part, so I showed there's a developmental switch, as we call it, in NPC composition that is absolutely essential for this differentiation process. And as I said, overexpression of Nup98 seems to accelerate the expression process, presumably by already expressing genes that have to be expressed down the road, but already at an earlier stage. There's an integral membrane protein that regulates the expression of those very specific genes. So it's not a general transport effect, if it is at all. It's a very specific one. So, but also we wonder, is this a transport-independent role, and what could that be? Is this a signaling step or whatever it is? And so since I don't have any molecular mechanisms to explain this specific nucleoporin, I want to point out in this slide some other interesting features that we found, which we can also not explain, but that segue into the part which actually has some molecular explanations, as you will see, and point to a direct role of nucleoporins in gene regulation. And one of the things that we noticed doing testing the mobility of some of these nucleopore components at the pore, so here we measure how long is a nucleopore component present at the MPC. We see that Nup50, for instance, changes. So here is very dynamic in myoblasts, so these are non-differentiated cells, but then becomes less mobile in myotubes. Curiously enough, although Nup50 is expressed both in myoblasts and myotubes, when we knock it down, it also blocks muscle differentiation. So again, it's another nucleopore component that is required for differentiation. If we knock it down in myoblasts, myoblasts don't care. So it's not an essential, again, transport factor. And Nup50 is one example that shows that behavior, but Nup98 is another one. And Nup98 got us really excited because there was actually a paper, there were actually two, I only cite the first one, where it was shown that the dynamic, doing the same experiments that I just showed you, people have done in tissue culture cells, and the mobility of this nucleopore component was somehow dependent on transcription. So when in this paper, they blocked transcription, Pol2 transcription, the mobility of this nucleoporin changed. And so we thought, well, okay, that's interesting, and maybe that would indicate that nucleoporins are involved in transcription in a much more direct way than via nucleocytoplasmic transport. Now, the idea that nucleopore components are involved in gene regulation is obviously not new, and with Jason here, I guess you all know that there are many papers that claim that, and shown that nucleopores can interact with inducible genes in yeast, and Jason's lab is active in that area. It also has been shown in yeast that nucleopores can act as chromatin boundaries. It has been shown that nucleopores can actually participate in looping of genes at the periphery. That's also in yeast. And looping by Nick Proudfoot's lab has actually been shown to be critical for stable expression. And there's been a few studies in Drosophila which links nucleoporins to Dorset's compensation and to hyperactivation on the X chromosome. And there was one paper where looking at global histone acetylation in HeLa cells, there was some, in ChIP-seq analysis, there was some interaction of the genome with the peripheral pores. And so we were wondering, well, do nucleoporins play a role in transcription in high eukaryotes? And we decided to focus on the Drosophila polytene system because it has been very successfully been used to uncover basic fundamental biological processes, such as chromatin modification and transcription. And it allows you at a cell biological level just to look at the entire genome in a microscope. And as you know, or many of you know, I think, polytene chromosomes are the product of endoreplication, and so you have 1,024 sister chromatids lined up next to each other, and so you can see open chromatin, which is actively transcribed chromatin. You can discriminate it to condensed, which is more heterochromatic, and also highly decondensed areas, which are called puffs, which are sites of massive Pol2 expression. And so the first thing we did is now do nucleopore components interact with the Drosophila genome. And the first stains we did were already pretty amazing. So here is Nup98, and you can see that Nup98 binds to very specific regions all over the genome. It's not only Nup98. We also find this classical MAP4N4, which has been always used as the peripheral nucleopore marker, gives a very specific binding pattern. Sec13, which is part of the COP2 complex, vesicle budding, but also an essential component of the Nup107-160 complex here, very specific binding patterns. Nup88, which is actually a part of the cytoplasmic filaments of the nucleopore, also very specific patterns. And 153, which is part of this nuclear basket, so it's a nucleoplasmic structure, gives also very specific interaction. But not all nucleopore comets do it. And I just want to point out that all of those interactions were verified either by hypomorphic mutants or by knockdown, so that we are sure that we're really looking at the specific sites and not unspecific interactions with antibodies. Now, these interactions of nucleopore components with the genome fall into at least two different categories. The first category, and I show you Sec13 here, co-localize with actively transcribing, so that is serine 2 phosphorylated Pol2. The other class, like Nup88, does not co-localize with Pol2. It never does during development. And we still have no idea what those sites are. We are in the process of finding out. But there's at least two, Pol2 and non-Pol2. And now for the rest of this part, I will talk about the Pol2 sites. We can also now use this polytene system to actually study development, because we know, and this has been worked by Michael Ashburn several decades ago, we know each gene that is upregulated and downregulated as the salivary glands develop. And the master regulator is ecdysone, which is a hormone that leads to the downregulation and upregulation of certain genes. So we know all that. And now we can ask the question, well, when do nucleopore components associated with genes? Do they associate before transcription starts, at the end? And are there multiple nucleopore components that would associate with a certain site? And this panel here shows you that Sec13 and Nup98 actually co-localize at sites. So it's more than one nucleopore component that binds to those sites. And here's a co-localization with actively transcribing Pol2. And doing those studies, we realized nucleopore components are there before Pol2 gets actually phosphorylated, pushed in a serine 2 phase, before you get the kick into elongation. So it's very unlikely that they play a role in transcriptional termination or anything. So a very early event. And doing those kind of study, we also realized that Sec13 is actually is one of the first ones we noticed. That's required to bring in Nup98. But then we find many others, like Nup50 and Nup62, which are actually required transcription elongation. So there's a stepwise assembly of a subset of nucleopore components. And I don't want to give you the impression that we think there are little nucleopores sitting there. This is not an entirely assembled pore, but there are parts of nucleopore of the NPC that in a stepwise fashion assemble at Pol2 sites. Okay? And obviously the big question is, well, it's nice if they associate, but are they actually essential for the transcription of those genes? And there we were very fortunate, because usually it's hard to do those experiments by knockdowns, because if you interfere with NPC components and you get a nice phenotype, you don't know if this is a direct consequence of the function of the nucleopore in this particular process, or if it's an indirect effect by perturbing nucleocytoplasmic transport. So that's always a problem. But in this case, we were very fortunate to have Sec13 as part of the nucleopore components of the Nup107-160 complex, because we have shown previously that this is an extremely stable nucleopore component and is actually not replaced over the entire lifespan of a cell. And I will come back to that very quickly at the end. So by knocking down, by driving siRNA specifically in this tissue for 16 hours, allows us to deplete Sec13 at this genomic loci, but leave the levels at the pore, because of its stability, essentially untouched. And we measure transport rates and everything. We cannot see any defect in NPC function. So we think what we're looking at, the phenotypes, are not a consequence by lowering pore numbers or interfering with pore function. Okay, so that's here. But so when we look now at the transcription level, the results were pretty amazing. So here you see a locus called 74EF and B. You see chromatin is highly decondensed. So these are one of these puffed regions. You see Sec13 there, but if we stain for 98, the other components would also be there. And Pol2. So this is again serine 2 phosphorylated Pol2. Now if we knock down Sec13, you see the chromatin decondensation is strongly inhibited or reduced. And Sec13 obviously is not there, but also the expression of those genes is strongly reduced. So that suggests that these nucleopore components are essential for the expression of at least those genes. And the interesting thing is, if you look at many other sites here, it doesn't come out very well, so there are many other sites that are not changed. So it's not that these nucleopore components are general transcription factors. They only hit a subset of genes. And this is also verified, obviously, by RT-PCR, because we know the genes that are in those loci, and also when we quantify the decondensation steps. So chromatin decondensation is strongly inhibited. The point is, it's only a subset of genes, and when we look at which genes those are genes that are either regulated by ecdysone, those are genes that have to be turned on during a developmental process. So there's a strong bias towards developmentally regulated genes, and somehow these nucleopore components are relevant for that process. Now let me come back to this first introductory slide where I show you that, and there's clear evidence in yeast that this is happening, that genes can actually go to the periphery and associate with peripheral pores. And so this system allowed us to address the question, are all those interactions that we see now of nucleopore components with the genome, do they occur at the periphery, or can they also occur inside the nucleus? And here we stain these large nuclei, which are about 50 micrometers in diameter. So those large salivary gland nuclei are enormous, so that's why the lamina is not really easy to preserve. But here in green, you see the nuclear periphery, and we stain with Sec13. And you can easily see while some of them are clearly associated with nuclear periphery, the vast majority is actually inside the nucleus. And during development, we know from John Sedat's lab that those sites don't move that much during development, so we believe those are actually remain inside the nucleus. And we have verified this for a specific loci with fish and co-stain with lamins. So we clearly see that many sites are inside the nucleus. They are not attached to the nuclear periphery. And that was important because that means that whenever, and there are several reports now where when you look at ChIP-seq data with nuclear pore components, the assumption is that those interactions always have to occur at the periphery. But our data clearly suggests that it does not have to be the case. And right, we also find this in S2 cells. So when we do a ChIP analysis, we find that nuclear Nup98, but that's true for Sec13 and the others, and we plot expression levels. So do we ask, are those genes or these targets of Nup98 actively transcribing or not? We find they are highly enriched in highly transcribed regions. So the same thing is not specific to this polytenized nucleus. It seems to happen also in non-polytenized nuclei. Now we have data also for ES cells, for mammalian cells, that the same thing is true. And the same thing when we look now, we ask, well, where are those genes? And compare, for instance, to lamin-associated regions, which have been shown, for instance, in Basque and Stainslet. So these are chromatic regions which are at the periphery. Yes, we do see some Nup98 binding sites that are at the periphery, but the vast majority is actually not in lamin-associated regions. So again, we think it's only a small set that is at the periphery. But we do think among these maybe 2 to 5% of genes, they're very, very interesting genes, even in Drosophila, but also mammalian cells that actually sit at the periphery. And those are very interesting genes which we're working on now. But for this talk, I think the majority is there are pore components that can bind the genome in a more global manner, but not necessarily having been associated with the periphery. And so that's the conclusion of this part. The NPC components directly regulate the expression of a subset of developmentally regulated genes. The majority of these interactions can occur inside the nucleus, and it's not at the nuclear membrane. And we also have evidence now, and we're wrapping this up, that Nup98 is also present in human ES cells and neuronal progenitor cells. So I think it's a general principle that those components regulate active genes. And I want to come back with now up to 10 pore components that bind to different regions in the genome. So I think nucleopores in general, not only Pol2 expression, but other chromatin organization events will become a very interesting part in the future because there are many things that we don't even know how they work. So which we have to identify where do they bind and what do they do. And I also want to point out Martin Forneroth's lab in Netherlands has published this back to back, and they come with a completely different approach to very similar conclusions. Okay, so with this, I want to move to the second part and talk about illegal border crossing. Since I'm in San Diego and you know there's a lot of discussion about the Mexican borders are closed, I think that's an appropriate title. And it also comes back to the fact that whenever, I think the nuclear membrane, as I said at the beginning, has been viewed as this passive membrane barrier for a long time. And whenever people see a barrier, then the first question that you ask is, well, how do molecules move in and out or across this barrier? So we know a lot about how these transport processes occur. And the first example that we found of an illegal crossing is something that we published 2 years ago now. And this is linked to a failure in nuclear pore function. And I want to show you only 2 slides, but as a warm-up for the main part of this session, which is not linked to pore, but with a nuclear membrane failure. But I want to point out, because it's an important point, that nuclear pore components are actually formed during embryogenesis. There's something very peculiar about nuclear pore components, and we're talking about the scaffold components. So when you look at one of these scaffold components, like Nup160, for instance, and compare it to Nup153, which was one of these peripheral, very mobile nuclear pore components, and look at it at its expression level, you find Nup153 is expressed throughout embryogenesis in every cell and throughout adulthood in every cell, although at different levels, but every cell expresses it. In striking contrast, Nup160 is limited to a few cells in the embryo, and those are actively dividing cells, and not at all detectable in the adult worm. Although the protein, if we just fuse GFP to Nup160, the protein is there in every nucleus in the adult animal. And that suggests to us that obviously all the nucleopore components here that you need in the adult have to originate from this early embryonic development. So a major conclusion from that is that they must be very long-lived, right? Because they are incorporated during embryogenesis and the cells leave the cell cycle. They no longer express this. I don't have the data here, but then they are no longer able to assemble. And we've shown this by many, many biochemical, genetic ways, cell biological ways, to be not only true in C. elegans, but also in muscle cells in mammalian differentiation systems. So the general trend is the scaffold components, as soon as cells leave the cell cycle, are downregulated, and then the scaffold essentially doesn't exchange. And we have no biochemical and cell biological evidence that they are really actually replaced. And this lack of, so they are extremely long-lived, as I said. They're built to last their lifetime. And the consequence of this is that there is a cost for this during aging. And now we're looking at neurons that are as long-lived as we are. So we have in our cortex cells that have to stay in a post-mitotic, in a non-dividing state for many decades. And so we ask in rats now, well, is there a consequence for the NPC function? And sure enough, we found the presence of tubulin, which should be only in the cytoplasm, and here are isolated nuclei, forming these large filaments inside the nucleus. And by many other data that I don't show you now here is that we found that the nucleopore deteriorates during aging. So some components are lost, and the permeability barrier breaks down, and things like tubulin can now leak into the nucleus, and then in some cases form this large tubulins, this aggregates. And why we're really interested in that process is because similar filaments have been seen in an age-dependent manner in many tissues, such as Parkinson patients, where you have your nuclei in the substantia nigra, you find very similar filaments. And we're working with a Canadian clinician who actually shows that in those cases you also have a disdain for tubulin B3, so neuron-specific form. So this would be one example where the permeability barrier fails as a consequence of the way nucleopore components are organized. And as I said, this scaffold structure is extremely long-lived, and we want to understand now how first this damage occurs in this long-lived cells, and if there are differences in the pore composition between a neuron and, for instance, a tissue that turns over. So that's ongoing research. But for the so this is loss of permeability barrier in the presence of active transport. I think that's an important point I want to make. From dextrin influx studies, we can show that in those cells, in this nuclei, transport is still active. So it's only a subset of pores that fail, but transport is still occurring. So the cell, there's no checkpoint that tells the cell, okay, well, some of my pores deteriorate, we have to do something about it. And the part that I want to spend more time now is another example which is not linked to pores, but which comes to the same, a similar consequence, where something happens, something dramatic, but cells don't realize that this happens. And that comes back to this permeability barrier. So I told you a major function of the nuclear membrane is to act as a permeability barrier. And it's the physical presence of the membrane and the intactness of the nuclear pores. And I just show you this one example that pores can fail sometimes. And the only time when the nuclear membrane is supposed to break down in metazoa is during mitosis. And this can be nicely visualized in this kind of long time-lapse microscopy experiments, where you see here GFP fused, there's a triple GFP fused to a nuclear localization signal, which nicely accumulates in the nucleus. And then when a cell divides, and we can see this by condensing of chromatin, you see it spills into the cytoplasm. And then only when you reform the nuclear membrane, you get the reaccumulation of this GFP into the NLS. So when you do look at a lot of cells, you can always see this spilling events of the marker. It's just followed by a mitosis and then a reformation. But then Dan Anderson, a student in the lab at that time, found something. Okay, well, the consequence, what I just said, of this is nuclear envelope breakdown. So the complete disassembly of the nuclear membrane. And while it looks as a catastrophic event here, it's actually a nuclear envelope breakdown. It's a highly coordinated process that involves the hyperphosphorylation of many nuclear proteins, such as lamins, and also involves the microtubule-dependent rupturing of the nuclear membrane, although there's debate whether this is really essential. And as I said, this is supposed to only occur in mitosis. And then what he found while he was studying, doing these time-lapse images, he found cases where this GFP NLS reporter would spill into the cytoplasm, but cells is not followed by a mitotic event. And something curious happens that this is actually re-imported into the nucleus, reaccumulates, only to spill again, gets re-imported again, and then a cell divides. Right? So that appeared to happen during interphase. And when we looked at this process in more detail, so by just looking at how fast do we lose this nucleoplasmic signal, we see it's a very rapid process. So we cannot actually dissolve it. It happens within 3 minutes. But then the recovery is pretty long. So it takes about up to 20 minutes to come from this state back to this state. Okay? And it's not only happening in U2S cells, which is the osteosarcoma cell line, but also in HeLa cells, also less frequently, and also in other cancer cells. Okay? Now, the first thing of what we thought, well, maybe that's a consequence of imaging. So, maybe it's phototoxicity or a consequence of overexpressing this specific reporter. And as far as we can tell, none of those can explain this phenomenon because there is no correlation with the frequency of this event with laser intensity, with wavelength, with expression levels, so we don't see any correlations. We don't think it's a consequence of imaging, and I will show you actually an example where we see this event happening in cells that have never been imaged. So here is a movie of one of those things and events, and so you here see GFP NLS in the nucleus, and you see this bursts of this nuclear marker into the cytoplasm, right? And you will see that this cell here and also this later on actually undergoes a mitotic event, divides. We have lost a few pieces here. And then the daughters also spill more frequently than cells that don't spill. Get this event. You see many cases like this. So this event occurs repeatedly in multiple cancer cells. Cells don't undergo apoptosis, so which means there's no program initiated that tells the cells, oh, something happened. I lost transiently my permeability barrier. This is actually the first example of a transient loss of nuclear permeability barrier in interphase. And we have only 2 cases where we observed a similar effect in fibroblasts. So it's very, very rarely happens in primary cells. But we do see it quite frequently in cancer cells. So what do we think is going on? Well, looking at images, time-lapse images, you can see in many cases that the nuclear membrane kind of bulges out very locally, and then we see the spilling of this reporter first very locally and then fills up the cell. So there seems to be some kind of stress, physical stress on the nuclear membrane, very locally. This is consistent with the M images where we see in those cells, where we see frequently parts of the nuclear membrane actually missing. Now, we don't know, obviously, if this is a result of this event, but we just can mention in those cancer cells, we see those events occurring. Now, we know that this is nothing to do with nuclear pores because we can mess up with nuclear pores no matter what we find. This cannot be induced by either preventing the assembly of pores or interfering with the assembly. So what we thought is, well, maybe there's some tearing, some physical forces that are applied to the nuclear membrane, which is consistent with this kind of looping out of or bulging out of the nuclear membrane. And something that would be consistent with the idea is the following observation, that the frequency of these rupturing events is higher in G2 than it is in S and in G1. Although it also occurs in G1, but much more frequently in G2. And if we treat the cells with low dose of nocortisol, which depolymerize microtubules, we actually see a reduction of the spilling event, which would indicate that microtubules play a role. But then the question would remain, how would microtubules associate or assemble in an interphase cell? We're not in mitosis here. That would allow such forces to apply on the nuclear membrane. Now, to test whether it's actually really cytoplasmic interactions with the nuclear membrane could account for that, we did the following experiment. So we expressed the KASH domain, which is a domain that is found in many proteins that specifically localize at the outer nuclear membrane and that can interact with inner nuclear membrane proteins such as SUN1s. And this KASH domain and these outer nuclear membrane proteins have been shown to interact not only with microtubules but also with actin and intermediate filaments. So those would be candidates of proteins that might link the nuclear membrane to the cytoskeleton and potentially some forces. And Didier Hotzig has shown by overexpressing this KASH domain linked to its transmembrane domain, you can essentially compete away these outer nuclear membrane proteins from the SUN1s, because the SUN1s, these inner nuclear membrane proteins, are chromatin bound, so they stay at the nuclear membrane. But overexpressing this domain, you essentially shave off these outer nuclear membrane proteins and prevent these cytoskeleton interactions with the nuclear membrane. So we ask, well, what happens if we express this KASH domain now in those cancer cells, and what's the consequence, if any, on those spilling events? And here's one of these outer nuclear membrane proteins on isolated nuclei, and if we overexpress KASH domain, unfortunately I have the panel here with actually showing that the KASH is localizing at the nuclear membrane, but we strongly reduce the level of these outer nuclear membrane proteins. And when we quantify the frequency of those rupturing events, we see that in the presence of this KASH domain, we see a strong reduction in the frequency of those events, which would be consistent with the idea that some cytoskeleton components, maybe microtubules involved, we're not sure yet, but some cytoskeleton components apply an abnormal force in those cancer cells that lead to the rupturing of the nuclear membrane, and then somehow also, which leads to the spilling of this reporter, and it can be reversibly, so there's also some mechanism must exist to reseal the nuclear membrane in order to reestablish the proper localization of those proteins. Okay? And why are cancer cells, or why might cancer cells be more susceptible than other cells? Well, one of the components we looked at was the nuclear lamina, which is an intermediate filament that stabilizes the nuclear membrane. And when we knock down now lamin B, the lamin Bs, there are two isoforms of those, in cells that don't show this phenomenon, we actually see in the knockdown a strong increase, like up to 80% show this phenomenon when we weaken or reduce the levels of certain lamins. Okay? So that suggests that's consistent with the idea of the lamina providing a structural mechanical support for the nuclear membrane, and losing that makes cells more prone to that phenomenon. And then we looked at lamin levels in cancer cell, which is actually not very well studied, and there are only two papers that describe aberrant or reduced levels in many cancer cells. But when you look at many, these are breast cancer cells, these are carcinoma cells, you see many cells have very, very different levels and very low levels of lamins. And so we wonder whether this might be one explanation why in cancer cells we see this effect more frequently. And also in these cancer cells, I think here we use this HCC2185. If we again overexpress this KASH domain, we see a reduction of this spilling event. So again, it seems to be the same mechanism and somehow linked to the cytoskeleton, but we have to find out what that is. Okay, so we see a transient rupturing of the nuclear membrane during interphase, which has not been seen before. It's transient, so it can reseal, which is pretty interesting because we're talking about a double membrane. We think it's a result of cytoskeletal forces and aberrant nuclear lamina, and those things have to work together. And this rupturing when it's resealed and the permeability barrier is restored. So that's why the cells don't die. They don't undergo apoptosis. If you have a terminal event, which happens in some viral infections, and you rupture the nuclear membrane, it's not reversible, then obviously the cell will die. Right? Now the big question is, okay, why is this important for anything, right? And the first thing that is expected, and we have seen for the GFP-NLS reporter, is the mislocalization of proteins, right? And we see nucleoplasmic proteins in cells where our reporter is mislocalized, while also nucleoplasmic proteins are mislocalized, right? And this is clearly different from a mitotic event where you see here chromosome condensed. We see a similar phenotype, but in this case you see chromosomes. It's not a mitotic event. It's like one of these interphase assembly events. But you can argue, well, okay, if you mislocalize this protein here, the nucleoplasmic protein, so for sure it has a nuclear localization signal or some other means to be imported in the nucleus. So the moment you restore the nuclear membrane, it gets reimported. So what's the big deal? So, right. And that might be true. But you might have to obviously think of cytoplasmic components, for instance, that leak into the nucleus and might turn on genes that shouldn't be there. But anyway, this is clearly a reversible step. But we found 2 cases where this phenomenon is linked to something that doesn't seem to be reversible. And the first is, in many cases, and I showed you the first example in the time lapse, we see this bulging out of the nuclear membrane. You see the GFP report is in there. But there's also histone. There's chromatin in this part. So here you see histone H2B. And in many cases, we see these bulges here floating off the nucleus. So, and you've also seen this in the movie. They actually float off and are released in the cytoplasm. So these are now micronuclei, which there's one mechanism to form micronucleus, obviously by chromosome missegregation. So if chromosomes don't make it into the spindle, then the micronuclei form. So that's one mechanism. But that could be another mechanism how micronuclei form. And now if these parts of the nuclear membrane actually contain chromosomes, parts of chromosomes, and if they don't have a centrosome, then a centromere, then they obviously are not inherited and are lost. So it's a potential cause for genomic instability. But the other thing that could happen, that you can envision, is actually what happens if those rupturing events are big enough that actually entire organelles can be trapped in the nucleus. And in many, many cells, and this is a normal fibroblast cells here, you find, for instance, mitochondria, but other organelles as vacuole very close to the nuclear membrane. And so we wondered, well, is there ever a case where actually an organelle gets trapped in the nucleus? And sure enough, when we stained the first studies we did with the mitotracker, we found dots inside the nuclei, and those dots had a high frequency inside the nucleus when we knocked on the lamina. We also found in cancer cells very clear presence with this mitotracker in cells that were actually spilling. But obviously these mitotracker things are nice, but they don't, you really want to see an electron microscopy image. And we're in the process of studying many of those cells that we treat, or also tumor cells or biopsies for the presence of mitochondria. But here's one example where you have actually a part of the nuclear membrane missing, as you can see here, and a single mitochondrion actually being present inside the nucleus. Now, when this is repatched here, when this reforms, there's no way this mitochondrion can exit the nucleus anymore because it's way too big to fit through nucleopore components. And obviously, as I pointed out, mitochondria are good in the sense that they generate ATP, but they also generate a large amount of free radicals, which are taken care of to some extent or to a great extent in the cytoplasm by enzymes like superoxide dismutase, which are absent in the nucleoplasm. So now we wonder whether the nuclear capturing of mitochondria, potentially other organelles inside the nucleus, could work as a mutagen and could actually cause DNA damage. And if you wonder, well, okay, that could all be, that's all tissue culture work, and is this really, really relevant for anything? I mean, I cannot tell you right now. We have to really look for those things in a tumor, in an animal, but also in people, obviously. But something that would at least suggest this could happen is when you look now for the presence of nuclei. So you Google nuclear mitochondria. You find the first paper you find is this here. It's a science paper from 1965. And it shows the presence of here are 3 mitochondria. There's something that seems like a vacuole. And this is now not a tissue culture cell. That's actually from a solid lymphoid tumor, which suggests that this can happen in tumors, in people. Obviously, we don't know whether the presence of those mitochondria is due to the phenomenon that I just described to. We don't think it's very unlikely that these mitochondria can be squeezed into the nucleus during nuclear membrane formation, because we've studied that great process, and not even ribosomes during nuclear membrane reformation end up at the internuclear membranes. It's a very tight process, so we don't think it is caused by an aberrant capturing of those organelles into mitochondria. And what I want to point out is that clinicians actually use the term open nuclear membrane, not to refer to a mitotic event, but actually to an interface event where they see, in many cases, ruptured nuclear membranes, and they use this to discriminate different types of cancers. So for instance, carcinoma and lymphomas, for whatever reason, have more of those than other types of cancers. And they actually use this for cancer, not only diagnostic, but also prognosis. So they can actually look things at open nuclear membrane as a prediction if this tumor is aggressive or not. So I think there's a lot of interesting potential to kind of try to link what we see, this very basic cell biological phenomenon, with something maybe that might be relevant for either for a diagnostic, but maybe even for a deeper understanding of tumorigenesis in general. And so I want to summarize this, the last part, the transient loss of cell compartmentalization. Again, it's another example. I've shown you this little bit of information about the pores. So something deteriorates, but the cells don't realize that something's wrong. There's no checkpoint control that sends the cells into apoptosis. Same thing here. This is a transient rupturing in interface. There might be an effect, a slight effect on the cell cycle length, but cells obviously don't stop anything. They just patch it up, undergo the next division, and lose in that either mislocalized proteins, which might be a problem if you have, for instance, a transcriptional activator compartmentalized in the cytoplasm, but allowed access to the nucleus might be a problem. But this is clearly a reversible process because those proteins can move in and out of the nucleus. Now, loss of genetic material, this formation of micro, that's clearly not necessarily a reversible process. And the presence of mitochondria in the nuclei could potentially also be quite harmful, although we have to do the experiments of it. So that's just the speculation right now. So I'd like to thank the people who did the work. So Jesse did most of the imaging work of the last part with Dan Anderson. And Maya and Yoon worked on the gene regulation. And where is he? Sebastian here is not in blue, but he did, together with Maxi, who has his own lab now at UCSF, did the Nup98, the first part of the differentiation story, and he will follow up and try to hopefully understand what this nucleoporin does. And so with that, I want to thank you and take any questions if you have any. Yes.
Do you think that requires any kind of nuclear breakdown, or is that a separate pool of nucleoporins specifically with these nucleoporins?
Yeah, that's a separate pool of nucleopore components. That, well, so we know that, so for instance, Sec13 is at the pores in this Nup98-160 complex. At the genomic sites, none of the other Nup98-160 components are there. So Sec13, but Nup98 is there. So Sec13, Nup98 is there in a different complex than the one at the pore. So that's our strongest argument right now. And obviously, we have now biochemically purified what's bound there, and we found some interesting components that either regulate, modify histones and participate in gene expression. So I think we're getting there to understand what they're doing. So they are not directly binding to DNA, so they're recruited to something that is there before, and it requires a certain histone modification, so we know that, but we don't really have it down to the mechanism. What does it really do? And it seems to be more involved in the elongation of transcription than in the initiation. But it's not the entire and the same pore complex or part of the pore, so that's our argument for that.
What do you suppose is the consequence of a flood of misprocessed, unprocessed RNA coming into the cytoplasm?
Yeah, someone actually pointed out, which I've never thought about, actually, during that time also ribosomes can obviously, import. So it's the other way. So you actually might have aberrant translation. But I agree. I mean, all this unprocessed mRNA that spills out can lead to aberrant translation. We have not looked at this at all, but that's something that, this is very early days. So this is, I'm presenting, this is the second time I present this, because now if we feel it's an interesting phenomenon, but now the major work is to show if it's really relevant for a tumor. I mean, even if it's not a key mechanism in actually during tumorigenesis, which I think, could be, for example, then I still think it's important to know that this can happen because we all use cancer cell models, tissue culture cell models, and that these things can happen, I think, have important implication for some of the conclusions that you draw for some of these very specific signaling pathways. So I think even in that context, it will be relevant.
Reports about nuclear proteins
Exactly. So that's one of the things. There's this, 10 years ago, Peter Cook had this nuclear translation, and there were Nature papers back and forth, and it's artifact. Yeah, certainly I think there's no barrier anymore, so it's conceivable that ribosomes, at least temporary, and I don't know what happens to a translating ribosome that would be captured in the nucleus, whether that can actually then, the ribosomal subunits can be exported, but the export is also linked to the biogenesis of the ribosomal unit. So whether actually a ribosomal subunit is aberrantly localized can actually get re-exported, I don't know. So that's another,
I'm interested in your result where you showed that treatment of cells with the cortisol
yeah.
And I was wondering if you tried Taxol.
We tried Taxol to actually see if we could stimulate this, and we didn't get any. So either we arrested the cells in mitosis, we couldn't do the experiment. So we never found conditions where we basically saw an effect. So it was very inconclusive. Motor process. So that's also the dyne. So we tried dynein, but then again, same thing. We have to do this again because I think that's so I think it's right to say, like, how can you know this more than anyone else. Yeah, microtubules obviously need to form and need some attachment point to actually apply force, right? And I don't know, but I don't know if anyone has studied. That's something we have to find out in these cancer cells, how microtubule dynamics actually are in those types of cancer cells that we look. Maybe they are abnormal. Maybe it has nothing. Maybe it has also involved in actin. So I don't know. So that's why we're so very careful with saying like what it really is. But we have this cortisol and the KASH domain experiment. We clearly, I think, suggest that some cytoskeleton component play a role in that. But what it really is, that's something we have to figure out. Yeah.
You speculate back to the dawn of eukaryotes.
Yeah.
Do you think nuclear pores arose out of some chromatin components? Or do you think that some of the nuclear pore components ended up with roles in chromatin and gene regulation as sort of side jobs?
That's actually a work from Mike Rout's lab at Rockefeller. So he looked at actually, so when you look at the structure of many of those nuclear pore components, they have very strong similarity with components of proteins that form vesicles, right? Like clathrin. And so his idea is that nucleoporins actually started out in evolution in the first eukaryote to stabilize these holes, the empty holes at the beginning. So there were empty holes that were formed by this, proteins that can either form a vesicle, but also this, pore membrane that you have to, when the ER engulfs the nuclear membrane, you have these empty holes that are stabilized with proteins. So that's his initial idea. And then slowly during evolution, then more and more proteins were added in that hole to make transport specific. Because even if you have a small hole of 50 nanometer, you get some sort of a selective barrier because things like big things cannot enter anymore. So you get some, but then during evolution, things have become more sophisticated and you allow signal-dependent transport. So I think that the chromatin function of Nup98, for instance, or this other, has arisen separately. So I think actually in some cases for Nup98 and Nup153, it might even be a misnomer to call them a nucleoporin. I think the ability to associate with nucleoporin might be relevant, but they're not. None of those components, although there are some papers that say Nup98 153 is important for mRNA export. But that's always with the assumption that they play an essential role in transport. You can also explain some of those data by a transcriptional role, right? So I think that's not out clearly when those evolve. But there's something very curious about Nup98, which is this one protein that we have evidence now from Drosophila, but also mammal cells, is a gene regulator. That's expressed together with Nup96, which is the most stable scaffold structure of the nuclear pore, as one large polypeptide, which gets autoproteolytically cleaved into two proteins. One gets incorporated in the pore. It's the most stable thing we can see. It's never changed, a structural unit. The other is this gene regulator. And those are together in yeast. So yeast cells have those exact same gene. It's a very unusual lineup for genes. It's one big polyprotein. That's conserved through evolution. Now, why is that? And that's a big puzzle in the lab that we've studied. So we have a regulator with a structural component. So as if there's a sensor going on, we want to measure something. So that's something we pursue. So it's very hard to, I think, predict when they evolve. But I think the chromatin stuff is either independently and then they were kind of incorporated in the pores, or it's a more secondary effect of a primary structural role to stabilize the pore. Does this answer your question, kind of?
I was going to ask, are those like Nup98 and other components on chromosomes, are they going through mitosis on chromosomes?
Yeah, that's another layer of complexity, that some of this actually go to kinetochores and are required for spindle assembly. So they also have a mitotic role and some of them, like some of the Nup97 complex members, 88, they are actually in the moment. So when cells move from metaphase to anaphase, I mean, you know this, probably have measured this, all those things. So in anaphase, you get actually you get a higher chromatin compaction than in metaphase, right? In mid-anaphase. And then there's this moment in mid-anaphase when you decondense, and that's the moment when you also bring in the nuclear membranes and form the nuclear membrane. And in that moment, there are some of the nucleoporins associated, not on kinetochores anymore, but they're distributed throughout the genome. And then there's this magic moment when they all move to the periphery. And it would be really cool to do a chip experiment in exactly that moment. But as you can imagine, that's very hard to do. Yeah, on kinetochores, yeah. Yeah. On kinetochores, also it stains the spindle in some papers. So yeah.